Okay, so it's my pleasure to introduce Ben Americus. He's a PhD student in Dr. Jerry Bartholomew's lab group at Oregon State University. In 2013, he outmigrated from his natal stream of Cordova, Alaska, to work with and study salmon in the Western US. He enjoys gardening and backcountry skiing in his current home of Corvallis, Oregon. Take it away, Ben.
Yeah, thanks, Nick. And yeah, thanks everyone for joining me today. It's an honor to be part of this seminar series, a lot of great speakers. So I'll just jump right into it here. So yeah, I'm a PhD student in Jerry Bartholomew's lab group in Corvallis, Oregon here. We study myxozoan diseases. Mostly I work with this one, Adenoma shasta, but these myxozoan diseases, they cause a lot of different fish pathologies. I included the hamburger gill disease that Drew Mitchell mentioned last week. He preferred the hamburger gill disease over this proliferative name. But there's a lot of other ones. Proliferative kidney disease seems to be this emergent pathogen in the West here. Cerebralis, notorious parasite, causes whirling disease. Yeah, and it's kind of interesting. Mostly I talk to salmon people in the West, but I know there's some catfish people in the room today. So yeah, I think these myxozoan parasites apply to all of us as fish pathologists. So most of this talk will focus on C. shasta, this native endemic parasite we have out here in Oregon and Washington. So I do a lot of my work on the Klamath River. In the river you have native salmon, you have these little polychaete worms. This isn't drawn to scale. These things are like the size of your fingernail, and these parasites are microscopic. But you have this weird life cycle that plays out in the river. So you have these actinospores, this myxozoan parasite. It'll attach to the fish's gills, swim up the intestines. And then over the course of about 3 weeks, it'll start to reproduce and proliferate in the intestine. And you'll start with these really nicely folded lumen here, and you'll end up with just this total soup, this no organization, not able to process food, the death of the fish. So after about 3 weeks, you'll see these little bean shapes start to appear in the intestines. And this is the next life stage of the parasite. And after about 3 weeks, if we infect fish in the lab and they end up with guts that look like this and leaky vents, kind of bloody, distended intestines, you'll get fish that die in the lab. So once the fish dies or these spores reach maturity, they start to be excreted, or they come out of a dead fish. And they're in the wild, for instance, in the Klamath River, they'll be dumped into the river and they'll settle to the floor of the river, the muddy bottom, and they can be eaten by these polychaete worms. And they live in these polychaete worms for about another month. I guess that's like the shortest amount of time that we know. So after about a month in the worm, you'll see these triangular guys start to appear here. And these will burst out of the worm's skin, kind of like a pimple, I guess, kind of gross analogy. Yeah, after about a month in the worm, they'll burst out, and this whole life cycle will repeat itself. So this whole thing infecting 2 different animals, going through all this trouble to destroy the intestine, the whole process can happen in a little over 2 months, which is pretty remarkable. So what I think is most interesting about this parasite is that you have these totally 2 different life stages. They look totally different. They infect 2 different organisms. One infects a vertebrate, one infects an annelid, but it's the same exact organism. Both of them are C. shasta. So myxozoans, the clade that C. shasta is part of, they're cnidarian parasites. So most of the time when people think of cnidarians, they think of things like corals, sea anemones, jellyfish, things that sting you when you're at the beach. Myxozoans are really, really distant sister taxa to this clade. Some of the recent studies say they diverged 650 million years ago. Really rough estimate, but sometime a long time ago, these things diverged from these free-living cnidarians that we know of. Since then, they've reduced their genomes, they've evolved to parasitism, they've become microscopic. A lot of changes have happened. And for people that aren't into phylogenies, I know not everybody likes looking at these things. Here's a nice comic to show what's going on. This was drawn by a student in 2018. And this group of myxozoans, these fish parasites, are getting welcomed to this party with free-living cnidarians, jellyfish, corals, anemones. And this was on the cover of Zoology Mag. in 2018. So we know that these myxozoans, these fish parasites, they're part of this phylum Cnidaria, and we're kind of interested in what they retained and what they've lost. So you look at a classic cnidarian, something like a sea anemone. It's got these nematocystis stinging cells. They're about 10 microns long. Then you look at sea shasta. It also has these stinging cells, but they're really small. It's like the whole parasite's shrunken by like a factor of 10. In this case, the whole organism is about 10 microns. And the stinging cells themselves, these little purple guys here, with these tubules coming out of them, they're only about a micron big. So this whole organism has been shrunken down, but it stayed looking really similar to the free living kind. And these nematocysts, they're ubiquitous in all cnidarians. They're really easy to spot in myxozoans just because myxozoans are so simple. You've got like sea shasta, Kudoa, you got all these different fish pathogens. And most of the time when people look at these fish pathogens, they're looking at fish infections. So these unfired nematocysts, these little coiled tubules here. Here you have a M. cerebralis actinospore, these things that cause whirling disease. It's got 3 of these nematocysts and it's firing one of them here. Really quick process, one of the fastest processes in biology. They say the acceleration of the tubules as fast as the acceleration from a bullet out of a gun. Kind of remarkable biology. But yeah, all of these myxozoan parasites, all of these different fish pathogens have these nematocysts. Let's highlight them here for you. I'm really ubiquitous. And they use them in a really interesting way. So you think of like a jellyfish or anemone, they'll use their nematocysts to like attach to a fish or to sting a person, inject venom, scare them off, or pull in a fish so they can eat it. These myxozoans use them in a totally different way. So this comes from a paper that we published with George Sisler in 2020. So this is a really zoomed in photo of a baby mountain whitefish, really like a couple days old juvenile mountain whitefish. This is a pore in its skin. And here you have Myxobolus cerebralis, this parasite that causes whirling disease. It'll float by and fire its nematocysts into this little pore, which is secreting a lot of mucus. And then it'll pull itself in. And once it's pulled in like this, it can burrow in with this sporoplasm. So this is the bit that actually causes the infection here. And this stuff will migrate into the parasite. And then once it's migrated in, you'll be left with this hole, and this actinospore part will just fall off. So it's really kind of unusual adaptation of this nematocyst organ. Some early work with myxozoans and also with free living cnidarians, they noticed that when these things attach, when these things discharge their tubules, there's a lot of calcium released. This kind of led to this hypothesis in like the late 80s, early 90s in cnidaria research that this whole process of nematocyst discharge is osmotically triggered. So these things are enriched with calcium. And then when they get the right trigger, they'll just dump out all of this calcium, and that allows them to fire the tubule. So a question, I guess I started on this myxozoan research in 2017, and the early question we had was, we prevent these infections, these myxozoan attachment with salt baths. So the rationale here is that if you have a lot of calcium that's stuck inside of this spore and it's released when it comes in contact with a fish, maybe you can just put a lot of calcium or another ion in the environment and it'll prevent the infection. So here you're just equilibrating the inside-outside osmotic gradient. And if you can block infection, you can block everything that comes after. If you can block attachment, you can block infection. And this might result in happy fish, uninfected fish. And it's kind of an interesting tidbit here, but the rationale for this research, this osmotic inside-outside bit, it comes from this sunscreen lotion that's produced by one of the co-authors on most of my papers. It essentially does the same thing with people. It puts a really salty coating on your skin. So if you come in contact with a salty nematocyst, it won't have any juice to sting you. So yeah, in 2019, we tested this with a bunch of fish exposures. So what we did is we took really small juvenile rainbow trout, and we took the parasite that causes the whirling disease, and we put a bunch of salt in the water, enough salt that we thought it would be able to inhibit the parasite, but it wouldn't impact the fish. And what we noticed is that depending on which salt we added, we could either make the parasite pre-fire its nematocyst so it couldn't infect the fish, or prevent firing entirely. And we noticed that a lot of these salts reduce infection. So here's just a little video of what this looked like really. So we had 3 fish for mixing in really small volumes of water, and each of these volumes of water was dosed with a different salt. So we did qPCR on these fish to see what the level of infection was. So in this case, qPCR, if you have a high number here, it's high infection rate, bad for the fish. Negative number, low infection rate, good for the fish. And the positive control is here. So you can see a lot of these different salts, sodium, potassium, magnesium, gadolinium, they significantly reduce the infection load to the fish. So I thought this was a pretty interesting finding. We published it in Parasitology. And I think it has some real applications for fish pathologists that are dealing with myxozoan problems. Maybe not in the Northwest. In the Northwest, it seems like most of our problems with myxozoans are infections that happen in the wild. And I guess a real problem with applying these kind of treatments is that it's okay to keep fish in like a low level salt for like 10 minutes, what we had for this exposure. But if you're like rearing something like a catfish or in high levels of sodium, potassium, magnesium, or I guess more specifically, if you're rearing rainbow trout, you'll encounter some problems with the fish, unintended consequences. So yeah, I started a PhD, kind of continuing on this work in 2019. And we've kind of had the ongoing question from my master's work, how does this host sensing actually work? Is it just the release of calcium? And is there a way to more selectively block it? So in free-living cnidarians, for instance, the sea anemone I've showed a couple times already, they've got a whole bunch of nematocysts. They've got these big complexes full of them. Myxozoans, M. cerebralis, it only has 3 of them. And these free-living cnidarians, these sea anemones, there's been a lot of really great research on them over the years. And they've really kind of dialed down the mechanistics of nematocyst discharge and sensing at like a biochemical level, which is something we're nowhere near close to with myxozoa. So for anemones, it's like a 3-step process for how they fire these things. First, they'll get a chemical signal. So a fish will swim by and some chemical on the fish will trigger this neuron to release this acetylcholine chemical, and that will kind of prime the nematocyst. And then the fish itself will actually trigger this little nidocil here, this pokey guy, and this will cause a mechanical stimuli to go through the nematocyst. And once it has these 2 signals, then these calcium channels will open up. And when these calcium channels open up, you get the release of ions out, you get the flood of water in, and this allows these tubules to distend and not infection to occur, but allows these tubules to extend and the nematocyst to fire. So yeah, basically we don't know at all how this works in myxozoa. At least we didn't know until a couple of years ago. We took a bunch of nice photos of these things. So this is basically a zoomed in photo of this with scanning electron microscopy. This is Myxobolus cerebralis. This is the unfired type. The fired type, we don't know at all about what the signaling is. At a really basal level, they found that different molecules that are found in fish mucus will cause these things to discharge, but they don't know what the pathway is for it, or if they have any of these steps. Something I noticed the other day is that as I was putting this talk together, is that the lips of these nematocysts, this little guy here, it looks a lot like it does between the free-living ones to the myxozoans. I thought that was kind of an interesting observation. So it's like a 3-step process in free-living cnidarians, but for this talk, I'll just focus on this calcium channel opening up, because this is really the process that drives this really powerful discharge. I think if you can stop this process, you can block infection. So these things are pretty, I guess, they're pretty intuitive structures. So you've got like a membrane-bound channel, you've got sensory devices on the outside and the inside, and you've got the central component here. Oh, sorry about that. And if you take the central component and you look down it, you can see there's this little pore on the inside, and this is where the ions will flow through. So in the last couple months here, I've done a lot of genome mining, transcriptome mining. You hear this referred to a lot in biology now. It's really not as hard as it sounds. You just take sequences you find on the internet from these really well characterized proteins. In this case, I was getting them from the sea anemone, and you look in the data sets you have available. So in 2021 now, there's really great transcriptomic data sets from like whirling disease, hamburger gill disease, Kudoa infections. There's really great transcriptomes from each of these infections. And when I say transcriptomes, I just mean the genes. We know what genes the parasite isn't expressing inside of infected fish. And two data sets I was really interested in looking at were C. shasta, so this fish parasite we have in the Northwest, and T. bryosalmonae, which causes proliferative kidney disease. And I guess the reason I was interested in these two data sets is that we have transcriptomes from both hosts. So we have the fish host and the polychaete host for both of them. So I'll show us some of these gene diagrams now. Basically, we know this thing from Nematostella, the complete gene looks like this. So we were looking for these genes in these different myxozoan life stages. I'll start with the life stages that come from the worm, because these are the things that actually go on and infect the fish. We found pretty complete ion channels here. We can do some phylogenetics on them, and we find that there's this NPQR type calcium channel, and goes in a bit of jargon here, but basically we know, if we know what the specific type is, we know this is a specific type here. We know they can be inhibited by these really unique neurotransmitter blockers. So SNX482 and omega agatoxin. These are like things that are found in like painkillers these days. They have like pharmaceutical applications. I think the next step in this work will be applying these things in these, seeing if we can block infection with these really specific pharmaceutical products. So secondarily, I looked in these data sets from infected fish, so the parasite that goes on to infect the invertebrates. And we found that these ion channels were totally lacking. They're absent in both cases. So it's kind of been an ongoing question for me, like, where is the rest of these genes? Do these myxospore stages, they even have these ion channels? Are they using something else to attach to the invertebrate state? Do they even need to attach to the invertebrates with nematocysts? They seem to have these things, but are they even using them? This finding raised a lot of questions for me. And I guess I was kind of curious, like, am I just crazy here? Is my data bad? Why is it that we're finding totally different genes between the same organism? So I looked at another protein that's found in nematocysts, nematogalectin, and we found that these things are totally the same between life stages. So in the case of C. shasta, this protein that makes up the stinging tubule here, nematogalectin, is totally the same between the actinospore stage and the myxospore stage. And the same was true for T. bryosalmonae. So I guess I run out of time here. My final point. Basically, what I've done and showed in this research is that we've looked at this myxozoan life cycle, and we've identified a protein that only seems to be expressed on one of the life stage that infects fish. And this raised some interesting applications for our lab specifically. We're involved with a lot of parasite monitoring on the Klamath River. We use qPCR to do this. So folks that are familiar with qPCR, you know it relies on binding to DNA. And this, I should point out, this monitoring is really closely tied to management of fish stocks in the Northwest. But yeah, this monitoring relies on qPCR. qPCR relies on DNA. And the problem with doing DNA on different life stages is that they have the same genome. So we don't know if this prevalence data is just this fish infective stage or both stages. So by identifying this protein here, I think we'll be able to package it into an assay that we can use to just detect this actinospore stage. So yeah, thanks. I know I've gone a bit over my time, but yeah, thanks to folks that have helped me move this along over the years and my funding sources. Yeah, and I'll take some questions if we've got time.
That was an excellent presentation, Ben. Thank you very much. Feel free, folks, to unmute yourself if you want to ask a question, or you can drop it in the chat box and I'll read it to Ben. Matt, I see your hands up.
Hey, Ben, I really enjoyed your talk. It was well put together. Very kind of cool stuff. I may have missed it, and I apologize if I did, if I caught in. What were the levels of salt that you were putting in that were causing that, erroneous trigger?
Yeah, it was a while ago I did those experiments. I think they were like, I think for sodium, it was like 100 micrometer, like was it micromoles? So yeah, really, they're really low. It didn't seem to cause any stress to the fish, but yeah, like really crazy low amounts. Yeah.
For those of us that are bad at math, you don't know that in parts per million, do you?
I don't know. I could send you the paper if you want. I think it has.
Oh, is it published? It's out there?
Yep, yeah, it's out there. Yeah.
Okay.
Yeah.
Cool stuff.
Yeah. I think I'd be really interested in if any of the catfish people can see catfish aquaculturists, if they can find any applications for that work. It's a little tricky to apply in the Northwest here.
You know, it's a different animal when you're dealing with flowing water versus static water, but that's kind of what I'm curious about, what kind of levels could cause that non-specific trigger. That yeah, cool stuff, man.
Thanks.
Dan, this is Jeff Wellbeezer. Well done, thanks. Is there just one ion channel, or could it be that there are others in the genome?
Yeah, I think there's definitely others in the genome. In that sea anemone, they identified that there was they actually like made them glow in different parts of the body, and they found that there's this one type that's specific to the nematocysts. So I think in the case of sea Shasta, we probably have the same thing going on. We have this one type that we've looked at that I've only really focused on that's specific to the nematocysts, but there's these other isoforms that are playing different roles. And it's a major question we have. Like, is this one I've been looking at, is it even related to the nematocyst? And I think to get that question, we need to do blocking experiments or make it glow with fluorescence.
Great, Ben, you got a compliment here in the chat about the beautiful EM pictures. They were great.
Thanks.
Another one, maybe this is a follow-up to Matt's question, but did you see any toxicity in those treatments that affected the fish?
Yeah, we monitored them for stress. I didn't see any abnormal behavior or anything. Our toxicity was just really me watching them closely. We were really conservative with those numbers. I see my advisor Stephen put that paper in the chat there, but yeah, we were really conservative with those numbers, like going well below the literature values for the salts.
Hey, Ben, great job on the presentation. I'm just curious, do you have any input on marine versus freshwater environments? Presumably marine environments can be rich in ions already. We have myxozoans that are in marine environments too. So do you think there's a different mechanism that's firing that polar filament?
Yeah, no, it's a great question. I think the difference between marine and freshwater free living cnidarians, like a hydra versus like a sea anemone, it's just like ion gradients. So it's not different molecular makeup, and that three-step process is the same between freshwater and free living cnidarians. My suspicion is that it would be the same with myxozoa, like Kudoa would work the same way as sea Shasta. But yes, it's worth testing, and I should probably look more into those marine myxozoan data sets to see if we're finding these same things.
Yeah, cool. Thank you.
Abby, I see your hand up. Oh, yeah, it went down. All right, Abby, if you have a question, maybe you can put it in the chat box. Any last questions? Okay. Just 1 minute left. Well, great. Thank you very much, Ben. Virtual round of applause for you. That was a great talk.
Yeah, thanks everyone.